Good morning, esteemed professors. My name is Zhang Tianshuai, and I'm from Naval Medical University. It's my honor to present here. My presentation's title is Proteomics and Metabolomics Characterization of Interstitial Fluid in Colorectal Cancer. Scientific studies have shown that tumor progression have closely related to tumor microenvironment, and it can be divided into three parts: cellular component, extracellular matrix, and the liquid import component, namely interstitial fluid. However, the interstitial component has rarely been noticed, though the interstitial fluid contains a large number of proteins and metabolites that exert pro-tumor functions. Therefore, we applied proteomics and metabolomics to explore it in depth, hoping to characterize the expression of proteins and metabolites in colorectal cancer tumor microenvironments from the interstitial fluid perspective. Here's workflow of our studies. We first collect tumor and normal tissue from 50 patients who underwent colorectal cancer surgery. Afterward, we applied centrifugation to obtain interstitial fluid from these tissue samples. Finally, we performed proteomic and metabolomic analysis on 200 samples in total, including tissue and interstitial fluid samples. We found that more differential proteins, approximately 3,200, and more differential metabolites, approximately 120, were identified in the interstitial fluid compared to the tissue samples, which indicated the interstitial fluid samples have more biological information. It is reported that tumor cell proliferation and the anti-tumor effects of immune cells are key factors in progression, and our geo-enrichment analysis of differential proteins in tumor interstitial fluid showed that the upregulated proteins were mainly associated with cell cycle pathways, reflecting accelerated cell proliferation, while the downregulated proteins were significantly enriched in lipid metabolism and immune response pathways, indicating suppression of immune function. We then performed the protein-protein interaction analysis, targeting differential proteins in lipid metabolism and immune response pathways, discovering that the lipid transport proteins were notably reduced in tumor interstitial fluid. Additionally, proteins regulating complement activation and innate immunity were reduced in tumor interstitial fluid. Furthermore, the upregulated metabolites in tumor interstitial fluid were significantly enriched in the Warburg effect, leading to proliferation of tumor cells. And in mitochondria, the ammonia metabolism is also highly activated, which has been reported to be associated with immune cell death. Overall, sorry about that. Overall, our study reflected immunosuppression and intense cellular activity within the colorectal cancer tumor microenvironment, and also prevent a research basis for enhanced tumor immunology. This is all my presentation. Thank you for your attention.
Now you have 3 minutes for the questions and answer session.
So you start with about 200 patients, right?
Actually, it's about 50 patients.
50?
And each patient we will to collect normal and tumor tissues, and then we apply the centrifugation to obtain the interstitial fluid samples. So each person have 4 kinds of samples, including tumor tissue samples, normal tissue sample, and tumor interstitial fluid and normal interstitial fluid.
Mm-hmm. So you have so that means you have a 50 person, right?
Yeah.
That enroll in this work, and you have about 200 samples that you get from this 50.
Yeah.
And because you are working with a proteomic and about the some that respond to the tumor, so you will have a lot of information about the annotation of the protein, right?
Yeah, we
And
got more
a lot of
information, but it's limited to a poster.
So my question is about how can you get a selection for candidate for making your workflow on the right hand of the presentation, because you already make a workflow for like a candidate protein for this one, right? Because you have at least 50 sampling, and that each one you have a different type of protein that relate, but which one is the one, how do you make a candidate for the all or about 50%?
Thank you for your preferential questions. Actually, when we make some data analysis about protein mix and metabolite mix, we will select the most. We will select the fold change more than 1.5, and the significant less than 0.05.
That means you use a criteria to select the protein, that selective protein, right?
Oh, I think it's a I think in data analysis we have to do this.
Oh, okay. Thanks.
Any more question from the online judges or here? Okay, please. Thank you.
Yes, from 50 patients, you obtain since the CRC itself has many stages and it can happen in many locations. Do you have any ideas roughly these genes proteins that you analyze correlates with a certain stage of the cancers or something?
Thank you for your professional questions. And as you said, it's about clinical data, and we collect all of 50% clinical data, and to do further data analysis. And unfortunately, it's not it has no significance about the protein mix and metabolomics data with the clinical data. So I didn't put it into our e-poster. Thank you.